Hello everyone, I am Dr. Desai, working as an assistant professor at PES Modern College of Pharmacy, Nigdi, Pune. So today we will see the technique called as hanging drop technique, which is usually used to study the motility of bacteria. Now following are the requirements for hanging drop technique. First is your clean glass slide containing cavity, which is also called as a cavity slide, cover slips, Vaseline or bites of paraffin, Bunsen burner, microscope, lens paper and lens cleaner, and 18 to 24-hour cultures of organisms. Now we will see the principle behind this hanging drop technique. Now usually what we do is we take a drop of bacterial suspension and put it on the center of the cover slip, and this cover slip is placed into the cavity slide in such a way that the drop is not burst, and the slide is observed under oil immersion objective lens. Now the bacteria are usually divided into two classes. First is the motile bacteria and the other is the non-motile bacteria. Motile bacteria are the bacteria which can move from one place to another with the help of flagella or any other organs for locomotion, whereas the non-motile bacteria, they cannot move from one place to another. So if the bacteria is motile, the cells will be seen to show erratic movement in the surrounding environment, and if the cells are non-motile, then they will remain static in the medium without any movement or may show Brownian motion resulting from the bombardment by the water molecules in the medium on the bacterial cells. So now we will see the procedure involved in this hanging drop technique. First, a cavity slide is cleaned properly under tap water such that the water does not remain on the surface. The cavity slide, it is a glass slide with small round depression at the center into which a small drop of bacterial suspension can hang. The slide is dried by wiping with bibulous paper and subsequently moving it over the flame or keeping it in sun for sterilization. A ring of petroleum jelly or Vaseline is applied around the cavity. The loop is sterilized over a flame and cooled, and then a loopful of bacterial suspension is taken from the culture aseptically, and a small drop of this suspension is placed at the center of the cover slip. The broth culture should not be more than 24 hours old because bacteria may lose their motility as they grow older. Now the cavity slide is inverted and placed on the cover slip. So here you can see this is a cover slip and this is your slide, and you are inverting this slide and placing it on the cover slip in such a way that the cavity covers the drop. The slide and the cover slip are pressed together gently so that the cavity is sealed. Care should be taken to see that no part of the cavity touches the drop. The slide is inverted quickly such that the drop hangs into the cavity without touching it. Then the slide is clipped to the stage microscope and the edge of the drop is focused under low power objective. Now, why is it necessary to focus on the edge of the drop and not on the drop? Better contrast is obtained due to difference in refractive index of the drop and the cover slip. As the drop hangs, it thins towards the edge, and for which the edge contains less number of bacteria to be observed clearly for motility. Usually, anaerobic bacteria come towards the edge to get more oxygen for respiration, for which they can be observed on the edge. And a drop of immersion oil is put on the cover slip just above the hanging drop, and the edge of the hanging drop is observed under oil immersion objective of the microscope. Now we will see the observation. Here you can see the bacteria moving from one place to another in this video. In this way, the hanging drop technique can be used to study the motility and morphology of bacteria. Thank you.